Dear colleagues, welcome to the OncoAlert Video Journal, bringing you the most talked about research of the week direct from the lead researchers. Today, we're talking with Dr. Renee Saliby, who is one of the main authors of a paper out on Nature Cancer, Immunogenomic Determinants of Exceptional Response to Immune Checkpoint Inhibition in Renal Cell Carcinoma. For context, the study investigates the molecular determinants of exceptional responses to immune checkpoint inhibitors in metastatic clear cell renal cell carcinoma. This is done by analyzing pretherapy genomic and transcriptomic data from treatment-naive patients. But here's Dr. Saliby to tell us more.
Hi Gail, thank you so much for having me on OncoAlert to talk about our paper that I co-led with Tejas Jamiat and Chris Abaki under the mentorship of Dr. Shweri, Dr. Shukla, and Dr. Braun. A lot of biomarker work has been done in the metastatic RCC field to try to understand response and resistance. However, what patients want is a treatment that eradicates their tumor for a very long time. And based on that, we created a definition of exceptional responses that incorporate these criteria and uses outcomes that we use in studies. So complete response, partial response, PFS, and tumor shrinkage. And our goal was to try to understand what are the molecular elements that mediate these exceptional responses. So we looked at whole exome sequencing and RNA sequencing from tumors at baseline from two phase 3 trials, Checkmate 214 that looked at nivo-ipi and Javelin Renal 101 that evaluated axitinib. One first finding that I think is very interesting is a higher clonal neoantigen load was associated with these exceptional responses. And this means that a bigger fraction of tumor cells that are harboring the same mutation was associated with these responses. And I think that makes total sense because this favors a good immune environment for the immune cells to attack the tumor. Another finding was that a higher expression of tertiary lymphoid structures, so TLS, was also associated with these exceptional responses. And these TLS are kind of structure aggregates of immune cells. And that again, we come back to a favorable immune environment inside the tumors. And when we also looked at patients who had exceptional responses but low TLS, here we saw that metabolic pathways were very highly expressed in these tumors. And from there, we created a signature based on these pathways. And we saw that patients who had exceptional responses expressed this signature very highly. And I think that's a very interesting potential therapeutic avenue because for now we're looking mostly at the angiogenesis and the immune environment. And metabolic pathways have been less studied in that field. So to resume, we found 3 elements that are potential biomarkers of the exceptional responses if they are validated. And also, we could think about increasing these mechanisms in order to enhance response to immune checkpoint inhibitors and achieve exceptional responses in all patients. Thanks again for having me, and please let me know if you have any questions.
Thank you, Renee. And there you have it, folks. Please feel free to follow Dr. Saliby out on social media for great kidney cancer research. And of course, access this paper direct from the Nature Cancer website through the provided link in this post.